welcome back. So this is week 6, lecture 5, so this is the last lecture of this week. So in this lecture we are going to move a little bit from our command line interface use and look at Genome browsers and look at two examples. One is your UCSC Genome browser and your IGV or Integrative Genome Viewer, and how this provides a GUI based interface to looking at all these data that you have analyzed and you're trying to visualize this data. So these browsers, how helpful they are. So we'll look at some example and walk through the use of these browsers. So before we begin, I want to mention that these kind of Genome browsers provide a very intuitive and user friendly experience where you want to look at the data and really see what the data tells you without having to quantify it in some way, because already the data is in a very graphical way and shown directly, and where you can just browse to that location and see what is happening in any given part of the genome. So UCSC Genome browser is again a web based interface which is quite popular and is very widely used when it comes to different type of model organisms which are hosted on this website. So if you go to the website, so this is how it looks like. It has various options to look at genomes, browse the genomes. It also comes with some tools which can do some operations on some of the data. And although this is the UCSC Genome browser is mainly hosted by University of California, Santa Cruz, versions of it are mirrored in different parts of the world. And apart from that, this is also available to set up on your own, right? So you don't have to just use the web interface. You can just download the source code of this browser and you can host it on your own server locally, or you want to host a different version of this browser, make some modifications, so that's also possible, right? So in the downloads, they also provide an option where you can download different data. So let's say you're doing a large genomics project where let's say you're generating genomes for large number of species endemic to your area, something like that. So you can set up a browser just for species of your interest and then host just those species on your version of the browser, right? Also your UCSC Genome browser provides you an option to add your own custom data, and you can analyze them along with the all data that is hosted by the browser. And various projects that are underway in UCSC, those are also available here. And a very nice help page is available which gives you a very elaborate tutorial of how to use it, what are the options that are available, and what are the new developments that are taking place and things like that. So if you are interested in using this, I would say this is a very good resource to get you started to understand all the intricacies of the UCSC Genome browser. So this lecture is not meant to be a entire course on UCSC Genome browser. It's more like an introductory lecture which tells you, okay, there is something like a UCSC Genome browser, you can use it for these particular applications, and these are some of the major tools that are available within the browser, right? So when we talk about tools, of course there is tools like blot which allows you to do alignment, right? You can rapidly align some sequences to the genome, see where they are aligning, things like that. Or you have a tool for doing in silico PCR where you want to design primer pairs along the genome, so you can do your in silico PCR. Or you have your table browser where you can browse to a certain part of data that is hosted on this table. Or you have a Liftover tool which allows you to convert coordinates. So let's say you have different versions of the genome, so you can just convert the coordinates from one version of the genome to another, or from one species to another. So that is possible with your Liftover tool. Then various other tools which are having a API, so that you can just use some programming language to access it. Also you have some variant annotation integrator. So let's say there are various variants in terms of single nucleotide polymorphisms. So you want to integrate them with other data, so you have tools for that. And this is the news page which keeps telling you what are all the new things that are coming up, whether it is new genomes or new tracks that are being added. As you can see, this is updated quite often, and new data sets are coming up on UCSC Genome browser all the time, right? So let's look at one example, that is the genomes, right? Because the major focus of your genome browser is the genomes. So several high quality genomes of many important species are hosted. So of course your human genome is there, different versions of the human genome are present, mouse genome, different versions are present. Then of course here the SARS-CoV-2 genome is also hosted. So apart from this, various other genomes which are of high quality of various model species, non-model species, C. elegans, Drosophila, yeast, right? All of these are again hosted on this. So there is also a genome archive link here. So this is various genomes which have been finished to a high quality, so they are also hosted here. So when it comes to the various genomes that are available, there is a large number of genomes covering various eukaryotic species which are hosted on the UCSC genome browser. So in this case we are going to focus on the human genome, and we are looking at the HG38 version of the human genome. So let's go there. So click on it, you can just select this, and then you come up with a browser interface that looks like this. So this is just like your internet browser, right? So in your internet browser, what do you have? You have a address bar. So this is similar to your address bar. So you can think of it as a address bar. And this is a address bar which allows you to browse the human genome. Which version of the human genome? The HG38 version of the genome, which was produced in December 2013. So this was the version that was available from December 2013. Of course, newer versions where the entire thing is completed is of course available. That is your T2T version of the genome is also available. But in this case, we will browse this. So if you want to search or browse the genome, you have to enter something in the address bar. So when we talk about addresses in the genome, right? So they don't have URLs, but you do have gene names. So you can browse to a certain part of the genome where a certain gene is present. So for example, if you type the letter CYP, it lists out all the genes which begin with CYP, right? So it could be CYP11A1, 11B1 and so on, right? So these are the different genes which are there in the genome. So you can just choose the gene that you are interested in, and you can just go take your browser there. Or if you are not wanting to search for a specific gene, you can specify the position, right? Position could be a chromosome, or chromosome along with some coordinates, or it can also be some search terms. So it may not be a gene name, but it can be a synonym of a gene or some other meta information that is associated with the gene. So that could be searched here, right? So then your browser will take you to that particular part of the genome, and you can look at that particular part of the genome in greater detail. So this just what is auto suggested by your browser when you start typing, but of course you don't need to choose one of them. You can just type in your term and go to that particular location. So here we have come to a gene called SHH. So in this we are looking at multiple types of data which are shown in different tracks. So what exactly is being shown here? So at the very top it's telling you that this is on chromosome 7, but where in chromosome 7 are we located? We are located somewhere between these two locations, right? So this is 155 into 10 to the power 6, right? And then within that you have 801 kb, and this is going up to 812 kb. So this is going from somewhere from 801 to 812 kb. So if you were to measure the distance from here to here, it's around 11 kb, right? And then there is a scale also provided here. So it's telling you that this distance from here to here is 5 kb, which kind of makes sense, because if this whole distance was 11 kb, so the scale is for half of that, or slightly less than half of that distance. So you know that this gene is spread over around 11 kb because the entire gene is shown here. How do we know the entire gene is shown? So these blue rectangles here, these are your exons. So then these are the 3 exons. These are the coding exons, and then you also have these smaller less wide versions of these blue rectangles. So these are the UTRs, right? So this is the UTR, and this one is the 5' UTR, and this is the 3' UTR, because the direction of transcription is from this side to this side, right? So you see the arrows that are shown, and these arrows are shown along the introns. So you of course have 2 introns and you have 3 exons, right? So you have 3 exons and you have 2 introns. Then of course this for the main isoform that is there for this gene, this is the sonic hedgehog gene SHH. But of course there are other isoforms for this gene which are shown here. So this is one isoform here where it doesn't include the first exon, it's starting somewhere internally, and then this is one coding exon, this is another coding exon, and this is a third coding exon. So this is isoform 1. Then you of course have this main isoform, this is your isoform 2. Then you have other isoforms which are there which may not produce a protein coding form, but they may be non-coding isoforms of this same gene, right. So you get all these different isoforms listed. Then below that you have your OM-IM alleles, those are SNPs which are labeled or which are annotated in the OM-IM database. So these are SNPs which are known to have some important consequences and which have been studied in various detail. So they can also be linked to some diseases, they could also be linked to certain phenotypes, right. So based on various studies. Then you also see that the expression level of this gene is also given here across 54 tissues. So what are these 54 tissues? So this is actually taken from a project called Tissue Expression Atlas. So this is RNA sequencing data from 54 tissues from a very large number of samples, 17,800 samples from around 1,000 donors, which is from version 8 of this project. So all of these measurements of the expression level of this gene in such a large number of tissues from such a large number of individuals, that is available. So if you want to go into that, there is a option to just focus on that particular aspect. Then it's also providing you ENCODE annotated regulatory regions or cis regulatory elements, that is your candidate cis regulatory elements, which stands for CCREs from various cell types. So those are also shown here. So for example, this is the first exon. So just below that there is a promoter that is annotated, and then there are various other regulatory elements also annotated. So again, you want to focus on these regulatory elements, that information is available as another track. So you have a gene structure track. So this is the gene structure track. So this is the first track. Then you have the SNPs track. So this is the second track. Then you have the third track, which is the expression track. Then you have the regulatory track or the enhancer track, that is here. Then of course after this you also have the conservation score. So this is the conservation score across 100 different vertebrates. So genomes of hundreds of vertebrates have been aligned to the human genome, and you quantify which parts of the genome are conserved across all of these different species. So the greater the conservation across these species, the greater is the conservation score. So this is estimated using a software called PhyloP, and you get this track which tells you the conservation score, right. So if you look along this track and compare it with the gene structure, you see that the exonic regions are the ones which are having higher conservation score. So this is again another exonic region having higher score. This is another exonic region having higher score. This is again another exonic region highly conserved. So wherever the exonic regions are present, the gene is having higher conservation across all these different vertebrate species. Then again, if you see further, so there is so this is your fifth track. So this is the conservation score. So then if you look at the subsequent tracks, you see alignments with various species. So for example, this is the alignment with the rhesus macaque, alignment with the mouse genome, alignment with the dog genome. Then you see that there are some regions in these genomes which is not available. So for example, this particular part of the genome is not available in the rhesus macaque, and actually that corresponds to a exon in the human genome. So either this exon is lost in the rhesus macaque genome, or there is a assembly error in this rhesus macaque, so this part is not assembled, right. And then you also see that there are some other parts that are missing, and then in the dog genome this part is missing, right. Then if you go further and further along, right, so if you look at species like chicken or Xenopus tropicalis, exons are still relatively well conserved, but the introns have very little conservation, right. So as you go further and further away in evolutionary timeline, these genomes are having lower and lower conservation compared to the human genome, right. Then this is your conservation track. Then you also have a track with the SNP database, right. So there is a standardized SNP database, that is your DB SNP. So all the SNPs which are annotated in this database are listed out here. Then apart from this track, there is also something known as a repeat masker track. So in the repeat masker track, what you have is all type of repeats that have been identified along the genome, whether it is transposons, whether it is some ALU elements, all these different type of repeat elements, or it could be some simple repeats, all of them have been identified and they are again available as a track here. So for example, you have one repeat here, one repeat here, one here, right? So if you click on this repeat, more details as to what type of repeat it is available. So if you look at this kind of a browser, what you see is various different types of data can be shown for a same region of the genome. So this is just few example data that have been loaded, right? So you can load any n number of omic data sets coming from various different assays and just look at how they correspond to each other across these different tracks and try to make sense of it, right? So this gives you a very visual representation of all these assays, what they found out when it comes to this particular part of the genome, right? But this is not to say that this is the only major focus of the genome browser. There are of course various other things you can do. For example, you can add custom tracks. Let's say you have generated some data, your own, developed your own assay, you have generated your own data, and you have this available as some tracks. That is, maybe you have it as a bed format file, or you have some kind of a BAM file, or one of the other formats, like maybe you have a VCF format or variant call format file. So then you want to add the track to this browser. So the way you do that is you have to add it as a custom track. So if you look below this various tracks, so there is a button that says add custom tracks. So if you just click on it, it provides you an option to add this custom track. Also, if you see further along, there are options to search for tracks. There are various tracks that are already available on the UCSC browser. So you can just go and search for it. If this is some dataset generated as part of the ENCODE project or some of these other mega omics projects, you will find those tracks already available here. You can just search it and include it. There are also options to change the size of this browser window, that is, you want to zoom into some particular region, zoom out. So those options are again available. But when it comes to adding custom tracks, you just click on this button. So then it brings you to this another interface within the browser. Then it's telling you that, okay, you're looking at the mammalian clade. In the mammalian clade, you're looking at the human genome, and among the human genomes, you're looking at this particular version of the human genome, that is the HG38 version of the genome, and you can add some custom tracks. So when we say custom tracks, they can be in any of these different formats, right? It can be big bed, it can be big interact, big PSL, VCF, bed graph, CRAM, GFF, GTF. So there is this huge list of different formats that is supported by the UCSC genome browser. So if you have data in any of these different formats, you can load it from your system or from some remote location, or you can directly paste it. So if you just want to paste it, if your file is very small, you can just paste the file's data here and you click this submit button, then it will load it to the browser and you can just browse to the region of your interest, and whatever custom data you have loaded will be available as an additional track in the same browser. Just like how we were looking at different tracks, we looked at the repeats that are there, the gene structure, expression, regulatory elements. So same way, whatever custom data that you have generated, you can just load that from this interface. But of course, let's say you have a larger file, so you can use this choose file option and you can choose the file from your local system and then submit it. Or you can also give a URL. Let's say your data is not small enough to have on your local desktop. So maybe it is hosted on some FTP server somewhere. So you can just give the link to that FTP dataset and then you can just load it directly from that server. So that option is also available in the UCSC browser. So this gives you a lot of flexibility when it comes to adding some other datasets which is not hosted in the original UCSC browser. Apart from the browser, of course, there is a table browser where you don't browse the genome, but you browse through the various tables that are there. So when it comes to the UCSC genome browser, one of the advantages with this browser is various types of data, linked with this genome are made available as tables. So when it comes to this version of the human genome, if you look at the tables that are available, there are various groups, and in each of these groups there are various sub tables, and in each of them there are various columns which have this elaborate information about different things. So for example, this is the mapping and sequencing group. In this there is a track called assembly. So what this table tells you is, it tells you all the details about this genome assembly. Where are the gaps in this genome assembly? Where are the back clones assembled? Which parts of the genome are having very high quality assembly? So all of this information is available in this particular table. And if you want to get this information for the entire genome, you can download it for the entire genome, or you can specify the genomic location or a subset of the regions where you want this information. So just for that region you can download it, right? And when you want to display it, either you can download it as a compressed file or as a text file. You can download just the sequence associated with it or just the table. So this whole thing can be output, and then you can download it and do subsequent operations on this. So this provides a different look at the UCSC genome browser, where you are not just browsing the data, but you can also download all of this data and you can do some subsequent processing or analysis on that type of data. So this browser also serves as a repository of large amounts of data. And we mentioned that there are various tools that are available. So we look at the Liftover tool as one example, but of course there are various other tools. As you can see here, when we go to the tools tab, there is a tool BLAT for doing local alignment, there is a tool for doing in silico PCR, then of course there is the table browser, among other tools that are available. So let's click on the Liftover tools, then it brings you to this interface. So what the Liftover tool does is, if you have some coordinates, so let's say these are the coordinates in the genome, so this is chromosome 4 and this is from this position to this position. So this is a position in the genome. So this is position in the HG38 version of the human genome. So now you want to know what is the position which corresponds to this location in the T2T genome, right? So you have this telomere to telomere assembly. So this is assembled in 2022. So you want to get the corresponding position in this version of the genome. So you specify the coordinates or the list of coordinates for which you want to do this conversion. You specify various parameters which can be left default for most cases. Then you click on the submit button. So then it will convert all these positions from the HG38 version of the genome to the T2T version of the genome. So this can be a quite useful tool, because you may have all the analysis done and all these kind of regions that you have in the older version of the genome. When the newer version of the genome comes along, you just convert the coordinates so that you can do all the operations with the newer version of the genome, right? So this again one tool that is quite popularly used using the UCSC genome browser. Right, now let's move to another genome browser that is there, that is your Integrative Genomics Viewer or IGV. So it has both a web based version and also a desktop based version. So this gives you advantage that if you want to use the desktop version, you can just start it up on your desktop, just look at the local data sets that are there in the your desktop or workstation, just load them up. Doesn't require a internet connection, right? So if you have some data which is confidential, you don't want to pass it onto the internet, right? So then this kind of a desktop browser provides you a good opportunity. But of course there is a web based version of this IGV also available. So in this example we are going to look at the web based version of the browser, but the desktop version of the browser is very similar. It's just that you start it up from your desktop. So if you want to look at the web based version, so you just go to the IGV website, then go to the desktop app option. So then you'll end up with an interface that looks like this. So again here the HG38 version of the human genome is loaded. All the chromosomes are shown. So you have chromosome 1 all the way up to 22, then you have chromosome X and Y, all the chromosomes are shown here. So if you want to go look at any particular chromosome, just click on it and then you can zoom in. So this is chromosome 1, so you see the entire karyotype of chromosome 1, where you have all these different colored regions along chromosome 1 which have been highlighted. So you can zoom in, zoom out, depending on what resolution you want to look at this particular chromosome. So if you zoom in further, you will start seeing the different genes that are there on this chromosome, right. So it's showing the exon structure, gene name, right, all these details are being shown. And depending on whether you want to look at the entire gene or part of the chromosome, you can set the zoom level. Right. So this one way which is very similar to your UCSC Genome browser, but the difference here is compared to your UCSC Genome browser where you are mostly relying upon genomes which are hosted on the UCSC website. Here it is very straightforward, very easy to add your own custom genome, right. So let's say you produce a new genome assembly, you want a browser for that version of the genome assembly, you can just use this IGV browser. So let's see one example, that is to look at read alignments. So we saw how to generate BAM files by using read mappers, right. So we did some read mapping in the previous lecture. So when you do read mapping, you get these kind of BAM files. So what are these BAM files? They are alignment files which tell you where each of the individual sequencing reads is aligning to the genome. So now this provides you an interface for visualizing these kind of BAM files. So this is a chunk of the chromosome, right. So this is the entire chromosome. So this is the chunk of the chromosome that we are focusing on. So when we zoom into this particular part of the genome, you can see the reads that are aligned. This is the BAM file, the alignment that has been opened up here. So this is, let's say, one read. So this is another read, right. So this is the direction of the read. So that's shown by this arrow here. So there are many reads which are all aligned to this genome here. Then you see that there are some reads which are of slightly different color than one here. So these could be reads which are having spurious alignments, right. So those are shown, but the rest of the reads which are in gray color, they are aligning with quite a high confidence to this part of the genome. So then just above these alignments, you see the coverage, that is telling you how many reads are being aligned to this particular part of the genome. So you have various reads that are aligned, you see many reads. You can scroll further down, you will see more reads that are aligned. And then in this alignment coverage, you also see that there are some regions which are having stacked colored bars, right. So for example, this bar here is having green color stack, or here you have a stack which is partly blue, partly red, and various such bars, right. So these are telling you about the alleles that are there which are different from the genome and what is their abundance. For example, in the case of the green stack here, it's telling you that there is an allele which is present in all the reads, and this allele is different from the allele present in the genome. Or in the case of this bar where you have the red and blue stack, it's telling you that there are two alleles at this location. So one is the red allele, one is the blue allele, and both alleles are rather commonly present among the reads. So half the reads probably have the red allele, half of the reads probably have the blue allele, and you have many such positions where you have single nucleotide polymorphisms. So this is shown at this particular level of zoom, right. So if you want to look at individual single nucleotide polymorphisms, that's also possible. You just zoom in. So if you zoom in further and further, so you see the level of zoom has increased to maximum here. So then you see the sequence of the genome. So this is the sequence of the genome, right. So this is the nucleotides that are there in this particular part of the genome, and then you still have the reads aligned to it. So you see all these reads. So the gray rectangles are the ones which are aligned properly, and then you have the spuriously aligned reads, one or two of them here. But then this is the region that we are interested in. So we said there is a stack, and then in the stack there are some which are orange colored alleles and there are some red colored alleles. So the red colored alleles are actually the T allele, and that's matching with the genome. So since it's matching with the genome, you don't see the those bases, but they are present here. So for example, here the read doesn't have a base, so this is actually a T. Here also there is a read with no base, so that's also a T. This is also a T. So wherever the read matches with the genome, it won't show you the allele. But the cases where there is a mismatch, so you have the G allele in many of these reads, right? So you have G allele in many of these reads, those are shown as a mismatch. So here you have actually a SNP where you have the alleles T and G. So it's a heterozygous site with the alleles T and G. But then if you see at some other positions in this browser, so there are of course mismatches. So there is a G here in this read, there is a here in this read, there is a T here, there is a C here. So of course you do have some bases which are different from the genome, but these are not SNPs. How do we know that? Because if it was a SNP, you would see them in a large number of reads. You would not see them in just 1 or 2 random reads. So these bases that we are seeing, which are very infrequent and which are present at very random locations, these are probably sequencing errors, right? Because of course there will be some sequencing errors when you have 80 reads at a particular location. So you see there are around 80 reads at each of these positions. So of course you will have few sequencing errors, but this particular position we can be certain is a single nucleotide polymorphism, because there is a very consistent pattern of 2 different alleles occurring in these reads, right? So if it were a sequencing error, it would be more random like this, where you would see different positions having different alleles, and it's not a stack of reads which having just the same allele which is different, right? But you see that in the case of this SNP, you see that there is almost a equal number of reads with the T allele or the G allele, right? So this way you can zoom in to the base pair level and look at the SNPs inside your read alignment. So this is also again possible with your IGV browser. So these are some of the applications of what is possible when it comes to your IGV browser. So let's wrap up this lecture with some common uses of genome browsers, whether it is your UCSC genome browser or your integrative genome viewer or any other browser, right? So I've just covered 2 types of browsers. Of course there are n number of other genome browsers that have been developed over the years, and depending on how easy or difficult it is, you may want to use the browser which you find suitable. But whichever browser you use, some of the common applications that you will see is you want to focus on some genomic region. Maybe you are interested in some particular gene, or your particular region of the chromosome is interesting, or you have some vague search terms, maybe you have some group of genes that you are interested in. So you can use this browser to identify these regions, visually go to that location, look at all the data associated with that region, and try to solve the biological question that you are interested in, right? So this is one of the most common uses, that is visualize specific regions. Another application would be to compare across tracks, right? So if you have data from different omics assays, maybe you have done some transcriptomics, maybe you have done some proteomics, you've done some epigenomic assays, maybe people have generated various types of data from some cancer cell lines, or there are some repeats, or maybe there are some enhancer elements or some regulatory elements that are identified. So all these different types of data are available, and you want to know how they overlap with each other. Are some single nucleotide polymorphisms located within the exons? Are those single nucleotide polymorphisms leading to synonymous changes or non-synonymous changes, right? So this kind of integration across different omics data, or even different type of tracks, that could be another application that becomes possible once you load all this data into a browser. When you focus on these specific regions, one thing you can do is you can take snapshots of them. So in many cases what you do is the snapshots themselves can illustrate a very important point, right? So they can actually lead to publication quality images by themselves, right? So that in that way these browsers provide very high quality options to export images as well. Or you could download some other information, because let's say you focus on some large part of the chromosome and you want to subset your larger data only to data in that sub region of the genome. So that's also possible with these kind of browsers. And as we saw in the example of the UCSC genome browser, or in the case of IGV, it's possible to load your own data sets, right? Not just one, but multiple data sets can be loaded. And again, for loading your own data, there are various options provided by these browsers, and you can link these data that you have, your own data, you can link them with other data that is hosted by these browsers and see how they compare with respect to each other. Then apart from the visualization option, or just the option to browse the genome, almost every browser comes with various tools that are associated with it, whether it is your UCSC genome browser or IGV. There are various tools which allow you to do different type of operations of different sophistication. Again, this varies from one tool to another, one browser to another. We looked at some tools like Liftover, and we looked at table browser, right. So these are the two tools we looked at in UCSC. Then again in IGV there is bunch of tools available under the option of IGV tools. So these can perform very different tasks which otherwise you may be doing using some command line tools. So those are already integrated into these browsers so that you don't have to do it separately, right. So this is again another use for your browser. So then especially when it comes to IGV, you have an option to write scripts, right, batch scripts, where you can take snapshots of the different parts of the genome, and you can do it for large numbers of genomic regions. Maybe you want snapshots of thousands of genomic regions. So to manually go to 1,000 regions, check thousands of regions can be very difficult. So if you have a batch script which can do this, which can just do this in an automated manner, that can be quite useful. So IGV provides you this option of running batch scripts for doing these tasks. So this is again another common use that these browsers have been used for, right. But this is not to say that this is all that browsers are used for. Of course, there are various other browsers as well which provide very specific applications that you want to cover. So we will not go into all of them. So the idea with this lecture is to introduce you to the idea of genome browsers and demonstrate what is possible, right. So we will go into these browsers in some more detail in one of the subsequent weeks. We will use the browser to explore some datasets. But for now we will end this lecture about browsers and continue next.